I'm making this screencast because for whatever reason during class on Friday, the iPad that I was using cut the video off after about 16 minutes. So I'm going to pick up where we left off there. At the end of the video on Friday, I was talking about the central dogma. We began talking about a gene, and I was trying to get across to the people in the class that we used to think of genes in relatively simple terms, a gene being a sequence of DNA nucleotides that codes for a particular protein. But what we realize now, and realize then to a certain degree, but we didn't kind of modify our definitions very well, is that DNA codes for lots of things. It codes for proteins, but it also codes for other things like RNAs, for example. So transfer RNA, messenger RNA, ribosomal RNA, those are all gene products, but they're not proteins per se. And so we now use the term, we use the definition that a gene is a sequence of DNA nucleotides that codes for a functional product, and that functional product can take on a number of different forms. The reason it's good to know about, have a good working definition of a gene is, first of all, genes are hugely important not only in animal behavior but in all the other subdisciplines of biology. And it's just one of those fundamental ideas that every biologist should know. But it also is key to the central dogma, because what we do in the central dogma is cells take DNA, the genes that are in DNA, they transcribe that DNA into messenger RNA. And so the DNA, the nuclear DNA, is a template for the messenger RNA, and then that messenger RNA serves as a template to specify an amino acid sequence in a protein. And so we can think about this at kind of the cellular level. We have a DNA molecule. Remember that DNA molecules are held together with hydrogen bonds. We spent a little bit of time talking about hydrogen bonds in class. Hydrogen bonds are these bonds that occur between molecules. They're relatively weak intermolecular forces, and they're weak because they're bonds between the partially positive hydrogen atom in a particular molecule, in a polar molecule, and a partially positive atom in an adjacent molecule. I use the example of water, where you have a partially negative oxygen that then can hydrogen bond with partial positive hydrogens in other water molecules. So the two strands of DNA here are held together with hydrogen bonds, which means that they're relatively easy to break apart and dissociate these two strands of DNA from one another. Then you take the template strand, and you use that template strand to generate a new molecule of RNA, whether it's messenger RNA or transfer RNA or mRNA, messenger RNA, transfer RNA, or ribosomal RNA. Once you have a messenger RNA molecule, there are sections of it that aren't actually going to be coded. These are referred to as introns. The exons are shown here in green, and so the cell excises out those introns and then stitches the exons back together. Those exons then are what are going to be the actual coding sequences. There's a cap on one end and a poly A tail on the other end, and this enables the messenger RNA to be exported out of the nucleus through the nuclear membrane into the cytoplasm. Once it's in the cytoplasm, ribosomes, there are two subunits to the ribosome. They basically will clamp down around the messenger RNA, and the ribosomes have active sites that facilitate the movement of a transfer RNA molecule into that active site. Each transfer RNA molecule has a particular anticodon at the bottom of it, and that anticodon corresponds to a particular type of amino acid. The other thing that ribosomes do, in addition to bringing those transfer RNA molecules into place, is it also catalyzes the bond between one amino acid and another amino acid to start forming an amino acid chain, which will ultimately become the protein. And so the transfer RNA molecule, each transfer RNA molecule is specific to a particular amino acid. And so we can see that in this case, the template strand with an adenine, a cytosine, and a cytosine is complementary to a uracil guanine in the messenger RNA, and that uracil guanine corresponds to the amino acid tryptophan. I believe that's what TRP is tryptophan. And so this codon will complementary base pair with an adenine cytosine transfer RNA molecule that then has the amino acid tryptophan attached to it. In the same way, 3 adenines complementary base pairs to 3 uracils in messenger RNA. The 3 uracils will then complementary base pair to 3 adenines in the transfer RNA molecule that carries around a phenylalanine molecule, phenylalanine amino acid on it. So I will skip forward, we can look at this in the codon table, and you can see these are the codons, which means a uracil would have an adenine anticodon, which corresponds to phenylalanine. Also notice that there is redundancy in the genetic code, such that in many cases all you need is 2 uracils, and anything can appear, 2 uracils, and if you have a uracil or a cytosine, that will specify phenylalanine. In the cases of cytosine uracil, any codon that begins with 2 cytosines will produce a proline regardless of what is in the 3rd position. This means that mutations that occur in the 3rd position are less likely to cause problems with the final product because there is this redundancy in the genetic code. And this brings us to mutations. Mutations are random permanent changes in the nucleotide sequence of an organism's genome or the extrachromal DNA and RNA. Substitutions, deletions, duplications, transposable elements, these are all the different kinds of mutations, but some of these mutations are more disruptive than others. Substitutions are less disruptive than deletions and additions. Deletions and additions are more disruptive. Duplications can be quite disruptive. And of course, transposable elements are large sections of chromosomes breaking off and coming reattached at different parts of the genome, and that can also be quite disruptive. The rate of mutation can vary widely depending on the type of mutation, the gene of interest, the organism, the position within the codon structure. All these things affect mutation rate. So the simplest mutations that we have are substitution mutations. These are referred to as SNPs, single nucleotide polymorphisms. There are 2 types of single nucleotide polymorphisms. The first of these is a transition. A transition is a shift from a pyrimidine to another pyrimidine. Remember that the pyrimidines are the single ring structures, or a purine to a purine. Purines are double ring structures. And these occur more often than transversions, where you substitute a purine for a pyrimidine or a pyrimidine for a purine. And the reason that transversions don't occur nearly as often is that when you substitute out a purine for a pyrimidine, it changes the distance between the 2 chains of DNA, and when you're going through DNA replication, the cell basically detects those differences in the size, the width of the molecule, and they will go back and fix that, or in that replication cycle. So like I said, mutation rate is really pretty low. 1 in a million or so is a good walking around number. But once again, it varies quite a lot depending upon where in the genome it occurs and the type of organism that you might be dealing with. Addition and deletion mutations usually wreak havoc with all of the codons after that mutation occurs. And the reason that this happens is because this results in a frame shift, and the next slide shows you this. So here we have the template strand, which is TAC, TTC, AAA, CCG, adenine, thymine, thymine. This produces adenine uracil guanine. Remember, the adenine uracil guanine is the start codon, which always corresponds to methionine. And then you have a stop codon down here. And this messenger RNA will specify methionine, lysine, phenylalanine, and glycine. If something happens and you get an insertion, there's an extra adenine in the template strand, that means you get an extra uracil, and immediately it transforms this codon that was adenine, adenine, glycine into a uracil adenine, adenine, which actually turns out to be a stop codon. And the reason that stop codons are stop codons is that they complementary base pair to transfer RNA molecules that don't carry an amino acid. And so you get to this transfer RNA molecule, it doesn't have an amino acid to add to the growing amino acid chain, and as a result that terminates the process of protein synthesis. Here you have a case where there's an adenine missing, so this third adenine in this sequence up here in the wild type, you lose that so that this codon now becomes uracil, uracil, guanine instead of uracil, uracil, uracil. It changes not only phenylalanine, but it also changes glycine, changing phenylalanine to a leucine and glycine to an alanine. And so this produces a protein, but it's not a protein that has any function. And so this is referred to as a missense. This would be nonsense because you have a gene, but the gene doesn't produce a protein product at all. This produces a protein product, but it's the wrong protein product, and more than likely it is not a protein product that is functional. And at that point, we were about to start getting into random genetic drift, and that was where we stopped. And so this kind of brings you guys who weren't in class up to where we left off on Friday. So thanks for tuning in, and hopefully there won't be any problems with my iPad on Monday. Let me know if you have any questions.